Welcome to the July episode of Map of the Month from Vibraint. So the purpose of this podcast is that we want to talk about how we can use whole brain imaging as a tool for combining different data sets. And today is a really nice day because the guest on the podcast today is Shigeaki Kanatani from Karolinska Institutet. It's the first to develop whole brain in situ hybridization. Technical challenge that was many years coming along, and you finally made it, solved it, Shigeaki. And maybe you want to give a brief introduction to yourself and your background.
Everyone, nice to meet you. I'm Shigeaki Kanatani from Karolinska Institutet. I'm working as a researcher, but also I'm a facility manager in imaging facility. But it's called Biomedical Imaging Core in Karolinska. Then I'm managing the light sheet microscopes and also spatial transcriptomics equipment. And then I supervise also tissue clearing techniques, but mainly iDISCO, and also we recently developed the TRISCO method. It's an in situ hybridization method, but as Jakob explained.
And just a brief teaser, just to describe the significance of your discovery. It was actually on the front cover of Science, right, 2 years ago. So really, I think it kind of revolutionized the way we think about spatial biology in the brain and other organs for that matter. But the fact, so iDISCO has been around for a long time, 10 plus years. It's been possible to stain with antibodies and things in the brain, but it's not been possible to do in situ, right? So I want to go a little bit back in time and explain about how you got around to doing the TRISCO protocol and kind of also maybe some of the challenges you had along the way, because I know there were many.
When we start to think about the in situ hybridization on 3D sample is publication, the first publication from Carl Deider's group. It's called the EDC clarity technique. But that paper is, I think, really has a big milestone of 3D imaging. Then we are very surprised about the possibility they showed about using in situ HCR technique on the big sample, because penetration speed of the probe is really fast compared to antibody. Then one of the bottleneck we had, maybe we are still having about antibody staining, is a time-consuming process. So for the for prepare sample, just one sample, it can take 1 month.
So we know this.
That's huge bottleneck. And also at that time, we didn't know so much. We include everyone, not only us. So much about the mechanism of penetration and then also tissue clearing part as well. So sometimes staining was done using 3 weeks, but in case of the clarity, it's also a difficult technique. So sometimes clearing failed, then all experiment failed. Then I was a facility manager, so I was scanning many samples from different users, and then I saw many sad incidents.
In general, protocol optimization is a huge
their case of just optimizing the protocol takes 2 years. Then I wanted to solve this situation. Then I focused on the speed of the probe penetration. Then at first I just combine simply with combining in situ HCR simply with iDISCO, then but it worked actually without doing anything. So then we published that method in Nature Biomedical Engineering 2020. That, but then, we noticed that sometimes experiment didn't work well, and especially for big sample. But we tried the same method on brain tissue, but sometimes like a penetration is bad, or sometimes tissue clearing is not enough, like a transparency is not enough, and also signal is sometimes low. Then we realized that we need to like solve those issues to stabilize the technique. But that's the starting point of TRISCO development.
And so what was kind of the, because I know we also tried, like probably in parallel with you, to do in situ, and it failed every single time, like no matter what we did, it failed. So what was kind of the big breakthrough that you had in terms of getting it to work?
But actually we haven't experienced such like a big problem, issue, because but when we try, we could see signal, some signal always. But that those signal was often very weak, and compared to immunostaining, especially. So, but that's what I don't know so much. So we think we simply improved our method. So, but I think penetration part is a huge, I think, like a big difference, because, and also it's kind of hidden issue. I spend a lot of time to understand why penetration was sometimes very bad. Then, and also it was just, how to say, it's a luck. I found that
Yeah.
it's a mechanism of the penetration. So.
Okay, so I think one thing I really like about the protocol as it is now is that you use this hairpin amplification, right? So there's a built-in amplification of the signal, which is, I think, one thing that is really missing in the immuno part, because there you always just have to rely on secondary antibodies.
Yeah.
So, like, can you maybe explain about how this amplification, like, how that improves kind of the signal detection?
So it's about the in situ HCR mechanism, right? Yeah. So in situ HCR is like a new generation of in situ hybridization technique. Then key part is it's use a small two type of metastable hairpin, but two type means those two type of hairpin hybridize each other when initiator sequence exists. So we first hybridize the initiator sequence plus messenger RNA hybridization sequence, then make a initiation sequence at the target. Then we apply short metastable hairpin. After that, then these metastable hairpin hybridize each other. So one hairpin open up with initiator, then open the hairpin also open up another hairpin. Then it's repeat. It will repeat this hybridization. I heard it's 500 times. Then the signal will be increased. Each hairpin have a fluorescent, like for example, Alexa dye or something. Then signal will be amplified more than 100 times.
And I think this is super important, right? Because I think for antibodies, if you have a primary and a secondary, you can only amplify each protein maybe five times with a fluorescent molecule. But here it's like 100 times, right? You can amplify the signal in theory. So okay, so I think one of the like really eye-opening things for me was kind of the the last bit where you go into clearing, because normally there's a gradient where you basically take your sample through like a gradient to take them into the methanol. And I know here was kind of one of the major things that you found to preserve the signal in the tissue. Can you maybe elaborate a little bit on that?
Yeah. So yeah, but still, I think but I decided to change the solution without using any gradient. I decided to directly transfer from one buffer to another buffer. For example, methanol to 5x SSTT buffer or TRIS to high concentration of methanol. Then, but that is to remove the risk of the RNS activation. But I still hear that I say too dramatic to do that.
Yeah.
But I found it works. Then, I think my recent paper show there's some risk of a big sample shrinkage or like a precipitation a little bit. But if so, in our hands, I recommended to use 80% methanol from trace buffer, but a
Yeah.
little bit more efficiently wash out trace HCl.
Now I know we have and that might become like maybe talking about challenges and stuff. I think one of the things that we find when we do the iDISCO is that the samples, they shrink a little bit more than or TRISCO compared to iDISCO, and that can be a little bit of channels mapping them into a reference brain. So I think it's definitely something to be aware about if you do subsequent mapping into an atlas, that the shrinkage might affect the mapping. One of the things I find like super exciting about the possibility of doing RNA is that so far we've been limited to basically antibodies that worked not only well, but extremely well to be able to work in iDISCO, right? So it has to be super high quality antibodies. But in theory, with the RNA technology, you can look at any gene. Have you have tried so many different looking at so many different gene expressions? Have you found like is there a sensitivity level where you no longer detect the probe, or is it like can you look at every single gene?
So far, So in theory, in situ hybridization could detect any gene, but so far but as far as we tested, like a success rate of detection is roughly 70% of the gene we tested. Then 30% of the gene shows, most of the gene is no signal, or a small percentage it shows non-specific signal. But in case of the non-specific signal, we could improve and there possibility to detect the signal. But in case of the no signal, then so far we have no idea how to manage that. Maybe maybe it's like a 3D structure of the RNA or protein binding of RNA, and also one possibility is very tiny RNA can be washed out during the experiment.
Yeah. So I know we have actually tried to use in situ probes to detect like RNA or siRNA or something, where we basically you want to detect like a small antisense oligo, and that is very difficult. And I think one of the reasons is that RNA is not fixed in the tissue when you do cross-link fixation. So the possibility of washing out very small RNA species is high, and that could of course be a problem. So one other thing I wanted to ask you about, because I know you've been working on this, is like multiplexing, obviously. So how many genes can you multiplex in the same brains, and can you do like several rounds of detection?
in case of the 3D, for example, as you mentioned, we have like a still issue of detecting all gene, right? So 30% of the gene, or can be more, it's hard to detect now. Yeah, I think we'd like to first solve those parts. And also doing multiplex need a lot of time now. For example, in case of the TRISCO whole protocol is 15 days long. Then if we just doing a multi-round, maybe 1 week, 1 week, one round per week, maybe. But I feel that's too long to do multi-round.
Yeah.
Yeah, so for now we are focusing on is speeding up the experiment time to speed up multi-round, and also we'd like to solve the sample size issue because like a TORISCO or like other like in situ method makes sample extremely small. But TORISCO is not so extremely small, but smaller than iDISCO. Then that's like that's also I think not good. And also, especially we need to think about like a green chemistry. Green means like a safer side.
Yeah.
Then using methanol is like it's I mean CMR, carcinogenic, so that's a risky chemical is like not recommended recently, and then our lab member doesn't want to use those process. Then I'm trying to make a safer protocol and faster and also more gentle protocol to the sample. Then, but that improves, of course, effort. Yeah, we can save the effort of multi-round experiment. Also, that open up the like a very good way to combine other technique, for example, spatial transcriptomics on 2D, for example, or mass spectrometry, or those things. Yeah.
One more question I have for you, Shigeaki, is like, what is kind of the possibility of combining RNA in situ with protein detection in the same sample? Is that possible?
Yeah. It's possible, and also it's also demonstrated by other group. So
Yeah.
so it's for sure. And then organic solvent clearing method really preserve protein and RNA. So after that, but we can do actually anything. like, but spatial transcriptomics is also possible, but that's shown Ali's group, right? DISCO-sec. Protein staining is also demonstrated by the Rockefeller group. It's Tatsuya Murakami. They published MFISH 3D. They demonstrated protein staining combination. So it's possible, but that part also antibody staining need to be done separately. So that's taking time. So
The sequential order that you have to do it in, do you have to do the RNA first and then the antibody, or can you do the antibody staining first and then the RNA? Or is
I
there any
think
rules?
theoretically both is possible. But in case of the MFISH 3D, antibody is the last experiment. In situ first and then protein later. But theoretically, in case we do in situ staining first, we should keep RNA intact during the immunostaining. But that can be a little bit challenging. So I think immunostaining later is the after in situ is for now the best.
I know that when you amplify the in situ signal, it's not doing super well in aqueous solutions, right? That's why you also have to do the fast gradient.
Yeah.
And for the antibody incubation, that's usually in aqueous solutions. So is there do you have to wait amplifying your in situ probes until you have done the incubation with the antibody, or how would you go about that?
Yeah, I think everything can be done after doing the in situ. So after you scan the 3D image of in situ, then the sample can be post-processed by immunostaining. So
Okay,
yeah,
so now maybe I misunderstood you a little bit. So basically, you would record the in situ signal before you do the antibody. So basically,
mm,
you just have the same brain,
yeah,
but after you're done scanning and imaging the in situ signal, you take it back into the immuno.
yeah.
Do the immuno in the same step as the in situ, or is that not possible?
That little for technically now challenging because in situ hybridization buffer contain formamide. That formamide denature antibody. So combination of that process is not so good. So there are some antibody very stable. It's maybe okay, but in case of the amplification step, it's like a 5xSCT. Yeah, so it's possible to combine. But
Yes, but your recommendation would be that you start by doing the in situ hybridization,
yeah,
you get your signal,
yeah,
then you image the brain,
yeah,
then you take that brain, you go back, you do the immuno, and you
yeah.
scan the brain again.
But that's I say. Yeah, I'm a very technical person, so my recommendation is please develop new technology by yourself. But if you don't want to do that, current technology is doing in situ first, then finish until scanning, and bring it to rehydrate and start immunostaining. But,
Yeah,
yeah.
yeah, perfect. So what is kind of is there a size limit to the kind of how big samples you can do? What is the biggest samples you have tested?
Yeah, thank you for asking that. So in our TRISCO development paper in Science, we tested the big sample more than mouse brain, but that is rat brain and also uh, ma- guinea pig brain. Uh, yeah, sorry. In Japanese, it's called mormot, but it's guinea pig. Sorry, guinea pig brain.
Yeah,
Yeah. Then, yeah, but we little bit surprised that guinea pig brain worked also. Yeah. So, staining doesn't have any issue about in situ. Then, but light seed it's a little bit challenging, but we could scan successfully. So, but so far, but guinea pig is also possible. Probably very large tissue more than guinea pig is also possible. But there are like a light seed microscope limitation.
yeah. Comes actually more microscope limitation than a sample size
Yeah,
staining
that's
limitation.
what I think.
Okay, so that sounds like the in situ probes are way more forgiving than the antibodies when it comes to penetration of the tissue, as you also mentioned in the beginning, right? Because they're way smaller, they are much better at penetrating the tissue, and therefore, like you can do bigger samples and antibodies, right?
Yeah, so yeah, DNA hairpin penetrate very fast compared to antibody. So in case of so when it comes to very big samples, then I think in situ is probably only way we go because the antibody doesn't penetrate it.
Oh, okay, so I think question for you. Like, what's kind of where do you see the technology going in terms of speed? How fast do you think you will be able to do it, and how many probes do you think you can multiplex? And where do you basically see in situ hybridization go for whole organ or brain imaging?
Yeah. So, but I think if we just extend our technology, like we can just do multiplexing with current technology level, then I think like a 20 or 30 gene detection in the same brain is doable. But of course it takes time, but it's doable.
Yeah.
Then I think 20 to 30 is probably enough to conclude most of the like a scientific question or even like a clinical question as well. So that part it's okay, but time is the bottleneck of this experiment. So yeah, but if we speed up the 3D multiplexing imaging, then I think like how say the revolution can happen because everyone can try the 3D multiplex image. But that may need a little bit like a different technology. The current bottleneck of one of the also bottleneck of this technique is amplification speed of in situ HCR. It's need overnight or at least half day.
Yeah.
But of course, if you are okay to like get the lower signal, then maybe 8 hour or 4 hour is maybe okay. But like adjusting that part is also challenging. If we can speed up that part, then it's easier to run multiplex 3D imaging. But I think but in the future already maybe people is doing multiplexing. Yeah, it's this like a 10 or 20 is already very close. Yeah, and also it's becoming common. Yeah.
And the question is exactly like you mentioned, like how many genes do you need in order to answer biological questions? And I think one of the things that we are super excited about your technology, if you combine the in situ hybridization with all the spatial transcriptomics from the LN and the ABC atlas and stuff, you don't need that many genes to actually separate many different neuronal subclasses in the brain. So I think at the end of the day, you could maybe with a very small subset of genes be able to separate a lot of different neuronal classes in the brain, which I think would be super exciting.
Yeah, that's true. And also, well, we can also expect the development of like a highly barcoded method as well. Then we can speed up more about the
Yes.
detection.
Yeah. So I'm super excited to see what you come up with next, Shigeaki, and I'm so happy you wanted to take the time today to talk about the TRISCO, which in my opinion is the biggest breakthrough in whole brain imaging for a very, very long time.
Thank you very
super
much.
excited about that, and thank you for sharing your map for this map of the month in July.
Yeah.
Uh,
Yeah. You know, you are also part of our publication, right? So
yeah.
thank you very much for your help.
Cool. All right. See you.
Yeah. Thank you for